last session of the day. Fourth floor.
No, it's that was important to have to get that back. Who loaded it?
I loaded, but I mean, there's another one here, but I guess the technical person maybe.
I have a lot at stake in this one. I know it's working.
Several minutes ago, there are two here. It's strange. But I uploaded.
I need to find that because that's confidential for me. Work I was just using that for someone.
Was it Bill, the guy that came over?
I think he will be back.
She goes on at the same time.
Yes, they have like, there's like 4 things.
Yeah.
Yeah.
So,
Yeah.
I have to assume that's
I would think so. It does actually.
Because my wife's in for 5 minutes. You can tell what's going on there. Catch up afterwards, I hope. Downstairs.
Oh yeah, absolutely. I'll be there. Bring the audience as well.
Yeah, please. No, instead he tag teamed me and said, Dave, stay.
I think we can do it. Yeah. It'll be informal. It'll be very informal.
Yes.
So welcome everybody. It's the fourth floor. It's the last session of the day. But hopefully the people who come here, you're going to enjoy the talk we give you, which is really about Arvinas, the setup of the company, the technology platform that we're working with, some of the clinical data, some of the early research that we're working on in oncology and neuroscience. And we have a whole team here for you to ask questions of. We may make forward-looking statements that may or may not come to pass. So the history of Arvinas is a really exciting one. We're actually in our 10th year. This is our 10th birthday this summer. And the work that we were focused on when the company was founded was actually from some pioneering work that Professor Craig Cruz at Yale had worked on, looking at protein degradation, looking at ways of hijacking the cell's natural protein degradation mechanism. And there's a seminal publication in 2001 that Craig and Ray Deshays and many others talked about. And several years later, Craig formed Arvinas in 2013. And that was the first company in the protein degradation space. And since then, there's been a whole series of firsts that the company's been able to manage along with Craig at the beginning and this great team that's been built over the years. The first PROTAC really to show oral bioavailability. The first PROTAC to show brain penetrance. The first compound to get into the clinic and phase 1, and then phase 2, and now in phase 3. So there's been a whole series of firsts for the company, proving the technology, taking it through proof of concept, and showing, as you'll see later in the data, real benefit to patients. So the technology itself is protein degradation, and here's a schematic. And basically, what we do here is we make heterobifunctional molecules. Craig called them PROTACs, proteolysis targeting chimeras. Heterobifunctional molecules. One end of the molecule, there's a ligand that binds to a target that you want to degrade. The other end of the molecule is a ligand that recruits a ligase from the protein degradation machinery. Those two ends of the molecule have a linker. They bring these proteins into close proximity, and ubiquitin is added to the target protein that you want to degrade. Once there's 4 ubiquitins, that serves as a signal for the protein to be dragged off to the proteasome, where it's degraded, broken down into peptides, and flushed back into the cell. Now, the really unique thing about that, apart from hijacking a natural cellular system, is the PROTAC's released. And it's released in such a way that it allows it to do iterative flows of degradation. So very, very different from inhibition, where you have an occupancy-driven event. This is a proximity-based event, and it's iterative, and gives you a lot of different functionalities. So we eliminate rather than inhibit. We disrupt scaffolding functions of target proteins, which is, again, very different from inhibitors. And we can approach classically undruggable targets because of this bringing together of proteins as opposed to occupancy-driven. So very exciting new technology, and over the last 10 years, we've proven that it works in various settings in the clinic. So right now we have one program in phase 3. In fact, as of today, I think we can say we've got 2 programs in phase 3, which is very exciting. Another 2 in phase 2, and a whole host of different programs in our early discovery pipeline, which you'll hear from the team. We have a whole series of different partners that have helped us along the way, most notably Pfizer, who is our partner with our ARV-471. And over this time, the company has grown quite significantly with over 450 people now in the company, a large majority of which is based here in New Haven, although we have been able to hire people external to that. So a very exciting stage for the company. And this is the pipeline as it stands today. So from that start 10 years ago with a novel technology, trying to work out the various principles of how the technology might work, we've now got significant clinical assets. Vepdegestrant, which is our ER, estrogen receptor degrader, looking at metastatic breast cancer. It has a host of different trials ongoing. Phase 3 second-line trial. We should have a phase 3 first-line trial by the second half of this year, and a whole host of different combinations of drugs to work with 471. And this is all done in partnership with Pfizer. Bavdegalutamide, ERV-110, is an AR degrader for prostate cancer. We should be in phase 3 with that asset in the second half of this year. And again, similarly, other combinations with 110, but also looking at earlier stages of the disease. And then there's a next wave of targets coming through our pipeline, both in oncology and neuroscience, and you'll hear about those today. And behind that, maybe another 20 to 25 programs in the pipeline. So a very robust pipeline. The company's grown to this late-stage portfolio with a line of sight to commercial, at least with Vepdegestrant, but also a very good supply of new targets and programs behind that. So with that, I'm going to hand over to Ron Peck, our CMO, and he's going to take you through some of the clinical data on our main assets.
Hello. Thanks, John. So let's make sure I do this correctly. Good. All right. So I'm going to start talking about, I'm afraid to pronounce it because I still haven't practiced it in public, Vepdegestrant. So this is, I'll probably refer to it as ARV-471. So this is our estrogen receptor degrader. And this is the one that, as John said, is in partnership with Pfizer. And one of the reasons that as a pioneer in this area that we took this program into estrogen receptor degradation and the other program in the androgen receptor space is that these were highly validated targets. I mean, the thing that's really exciting about PROTACs is that they have the promise of hitting and addressing upwards of 90% non-druggable targets. So this was an important molecule for us to develop to, number one, prove the concept of a PROTAC, and then number two is that there was very much a high potential that this could, in fact, turn into a useful drug for patients. So the rationale is, again, that estrogen receptor drives a good proportion of breast cancer. 80% of patients have ER-positive tumors. And obviously, the medical need in this in breast cancer speaks for itself. 1 out of 8 women have estrogen receptor positive or have breast cancer, and then 80% of all breast cancers are estrogen receptor positive. So this drug, could potentially meet an important need. It turns out that this is the one tumor type where there actually is a degrader that is on the market. Fulvestrant is a drug that works primarily by degrading estrogen receptor, but it's a very different mechanism of action. It's not as, let's say, as elegant as a PROTAC, where a PROTAC hijacks the cell zone machinery for degrading proteins, the ubiquitin proteasome system. Fulvestrant binds estrogen receptor, causes a conformational change in receptor, and then there's a downstream degradation effect. So it's actually it's a sort of a moderate degrader. Somewhere in humans, it's somewhere between 40 to 50% ER degradation. But even with that, fulvestrant has largely been considered the most active therapy to target the estrogen receptor. So we already had a proof of concept there, and preclinically, 471 is a much, much more potent degrader, not surprisingly by its mechanism of action. So we had a very strong rationale as we were starting the program. And so there's a lot of information here. I'm going to walk you through it. These are the key data that we have from phase 2. So what's important is that when we started our trial, our phase 1 first-in-human study, we had to test the drug in a population of patients who pretty much went through all established therapy and progressed through it. So they pretty much were running out of treatment options. And as you see in the pink above, this is how much prior therapy these patients got by the time that they had gone on to treatment with 471. This is actually data from our phase 2. So it was actually a seamless phase 1-2 study, all in one trial. And you can see that 100% of patients got what is a class of drugs referred to as CDK4/6 inhibitors. This has been one of the biggest advances in ER-positive breast cancer treatment, with the first drug being approved in 2016. And then fulvestrant, as I mentioned, 80% of the patients had gotten this. And then lastly, metastatic chemotherapy was used in almost half of the patients. So this is, by all measures, a population that has really developed a lot of resistance. And there's actually literature, molecular profiling data that shows that as few as just a third of patients still have tumor that is driven by the estrogen receptor pathway. Even with this, the primary endpoint of our trial was clinical benefit rate. We saw a 38% clinical benefit rate. What that is either a confirmed response by the standard tumor measurement criteria or durable stable disease, meaning the patients had not progressed for at least 6 months. And that's pretty important because patients coming off of these drugs will progress on average within about 2 months. So 6 months or longer is very important. And we had 40%, which is pretty good given that only about a third of these patients have tumors that respond to estrogen. You can see the progression-free survival results. This is also this is when we compare to other treatments that have been tested in this setting, this holds up quite well. Again, fulvestrant has a median progression-free survival of just 2 months, and it is the standard of care in patients like this population. And then the other thing that I think was really important for us is that 471 was really well tolerated. This is especially important for breast cancer because as we move the treatment in earlier settings, patients can be on treatments for as long as 5 years at a time when you think about postoperative treatment, which is one of the areas that we're very interested in going in. And it also, as a test of the platform, it was really important for us to show that this is a very safe modality. So as John said, this is now in phase 3 testing. This is one of the phase 3 studies. So we're actually going against fulvestrant. And just for you to know, fulvestrant has a clinical benefit rate of about 10 to 14% in a similar population. So we have a lot of confidence in this phase 3 study. The details are listed there. This is being executed by Pfizer and has been, I think will be potentially show real promise for this therapy. This is looking at the number of trials that we have in this setting, a different kind of format as what John presented. But essentially what we're doing here is not just the one trial John mentioned that we just are starting a second phase 3 study. That's being done in the frontline setting of metastatic disease in combination with the Pfizer drug palbociclib, which is one of the CDK4/6 inhibitors that are used in standard of care. But also we have interest in going into early breast cancer in phase 3, as shown in the lower right. And we do have an ongoing early breast cancer trial looking for the effects on biomarkers in tumors that have been resected. So that's the 471 program. And then just touching on the other major disease area that we're tackling with our AR PROTAC franchise, and that is prostate cancer. So obviously another very important area of need. This is the most commonly diagnosed cancer in men. It happens in 1 out of 8 men. It's the second leading cause of death, second to lung cancer. And it's also another setting where there's well-established validation for the androgen receptor in prostate cancer. Androgen is a driver of cancer growth for prostate cancer. And we have 2 PROTACs here. Our first PROTAC, which was actually the first one that was put into the clinic for Arvinas, is bavdegalutamide. That's 110. I can pronounce that much better since I've had about 1.5 years of practice for that. And then we have a second PROTAC that has some different qualities, properties, 766. And as we look at these 2, we're actually thinking of these more as a franchise. We do believe that even just within prostate cancer alone, but even in other diseases that are driven by AR, that these 2 drugs could potentially coexist in the marketplace. So this is just a schema to look at the natural history of advanced prostate cancer. On the far left is the latest line setting. These are the patients who have already gone through all established therapy. They've already gone through a number of different androgen receptor treatments, and this is a very unfortunate population where there's very little treatment that's available to these patients. This is actually the population that we did our first in-human trial, our phase 1/2. So we'll talk about the data in this very highly resistant setting. And then as you go into the earlier settings, it actually becomes much more dependent on androgen receptor. So just like the story with breast cancer, where the more treatment you get, the more resistant you are to estrogen therapy treatment, it's the same thing here. For both of these programs, we're very interested in getting into earlier and earlier populations. So what I'll do is the phase 2 data are shown on the left. If you can just focus on lower left, if you can just focus on that. So these are phase 2 data that we presented about a year ago, 1.5 years ago. And we will have another update in our program towards the end of this year for bavdegalutamide, and we'll have actually data for 766 later this month. But even, as I mentioned, even though we did our trial in a very, very resistant setting, one that is highly known to be have a high level of AR-independent resistance, things like P53 mutations, things that are pretty common mutations in oncology, we still found a population that had a pretty remarkable activity level. The way that we look at activity of prostate cancer drugs in metastatic disease is to look at PSA reduction. So of course, PSA is what's used to screen for prostate cancer, and if you get a reduction of at least 50% from baseline, that's considered a response. So in a population of patients who actually were marked by the presence of mutations in the androgen receptor, these patients seem to respond the best. In other words, these seem to be markers of retained AR dependency despite all the other mechanisms of resistance in other populations. And in this population, almost half the patients had a response by PSA, and we also showed tumor responses as well, in some cases long-lasting. And on the safety side, it was also quite well tolerated. Oncologists are used to drugs that have a high level of moderate to severe toxicities, and this was quite well tolerated. And discontinuations were quite uncommon, and dose reductions were quite uncommon as a measure of its tolerability. So this will be my last slide, and it just talks about the program going forward for both bavdegalutamide and 766. So bavdegalutamide is now really kind of teed up to start a phase 3 study. We've gone through health authority feedback with the FDA in Europe, and we have said publicly that we're targeting the end of this year to start another phase 3. It's pretty remarkable for a small company to have 2 new chemical entities starting phase 3 within about a year. We actually kind of did some homework and showed that we were on a short list of biotechs there. And then we also are looking at now combinations. It's a pretty common strategy in oncology. So we're looking at combination with the AR other targeting agent, there's a number that are used now as part of standard of care in earlier populations. Abiraterone is one of these drugs. It causes a reduction in production of androgens out of the adrenal glands. So you hit the AR axis from two different approaches. And we also have really interesting preclinical data that supports this combination, and we have an ongoing trial there. And then 766 is the second drug. As I mentioned, that we'll present some preliminary information about that trial, that phase 1 trial, before the end of the month, and we have started phase 2 already on that one. And then we're very interested in going back into this more in an earlier population where we can really show the promise of this treatment in patients who don't have the issues with AR-independent resistance. So with that, I think I hand it to Ian.
Yeah, Ian, please.
Thanks, Ron. Good afternoon, everyone. So I'm going to talk about what's coming next. We in the research group have been really proud to have the first PROTACs in the clinic, but we're not resting on our laurels. So I'm going to talk about two programs that are really next up, and then Katie and Adam, two of our very bright scientists, will be talking about two other programs. And these are really just a slice of our overall pipeline. John commented on it. I'm going to try to use this fancy feature here. Look at that. Oh, nice. We have 20 preclinical programs, over 20 preclinical programs in our pipeline. And we have 4 first-in-human studies being planned for the next 24 months, 2 years. So our goal really is to keep Ron very, very busy, keep him out of trouble, keep him off the streets. And these programs are both in oncology and immuno-oncology, as well as neurodegeneration, which is our 2 main therapeutic areas. And these are the product of our platform. And when I say platform, it's beyond just making the PROTACs. It's really understanding how to optimize them. It's understanding the trimer complex, the formation of the protein coming together with the E3 ligase that John described that's facilitated by the PROTAC. And with those type of things, we use structural biology. So we do crystal structures, cryo-EM. We do a lot of computer modeling, molecular dynamic simulations. How does that trimer complex form? How do we optimize it so that the ubiquitination is as efficient as possible and degradation is as efficient as possible? And of course, what reference to platform would be complete without a mention of artificial intelligence. It's not a gratuitous reference. We actually do use artificial intelligence. We generate a lot of data, pharmacokinetic data. We have a lot of compounds, trying to understand how to make these molecules as drug-like as possible. Because when we started, I can tell you, there was a lot of skepticism, because these are on the bigger side of small molecules. There was a lot of skepticism that we'd ever be able to make these things into drugs. We've obviously proven that with the data that Ron showed you. Oral drugs on top of that, and then crossing into the blood-brain barrier. So all that work, that platform fundamental work, has come into generating and making a sustainable, and what we think is a differentiated pipeline relative to the industry. I think we do have the deepest and most diverse pipeline of any of the protein degradation companies. We have a little bit of advantage because we've been the first ones, we've been around the longest, but nonetheless, I think because of the investments that we've made in the 10 years, really the fruits of all that are showing, not just in the 3 compounds in the clinic, but the ones that we're going to describe to you that are coming next. So the first one I'm going to talk about is called BCL6, or B-cell lymphoma protein, or gene 6. It's a first-in-class therapy for diffuse large B-cell lymphoma. So what is BCL6? Well, it's a transcription factor. It's actually a repressive transcription factor. I'm going to try to use this thing again. And so when naive B-cells have an antigen exposed to them, obviously, they want to generate a lot of antibodies to be able to help with that insult. And usually, basically, it stops the differentiation of the B cells to allow that hypermutation to occur in the immunoglobulin isotype switching. And then normally, after it's done, it turns off. And then basically, you get a memory B cell, plasma cell, and the apoptosis of cells that are not useful. So there's differentiation that happens. But if something happens to BCL6, it gets mutated, overexpressed, that doesn't happen. And so that oncogenic version of BCL6 does not turn off. And that leads to proliferation, inappropriate proliferation and survival of these B cells, and that leads to what's known as diffuse large B-cell lymphoma. So because it doesn't turn off, you can imagine then it's a perfect PROTAC target. We want to get rid of it. It's mutated, it's going, let's degrade it. That'll then turn off the whole system and then kill the diffuse large B-cell lymphomas. It's a transcription factor, so it's very difficult to drug. There aren't very good inhibitors. There's some out there, but they don't work very well. And transcription factors are difficult because not a lot of nooks and crannies for small molecules to bind to them. But with a PROTAC, you don't actually have to bind very tightly or very long. You just need to bind tightly enough and long enough to form that trimer complex to get to ubiquitination and therefore degradation. And that's the differential biology that we always talk about. Why is degrading better than any other modality? Why is it better than inhibiting? In this case, we have great data. I'm not going to show it to you, actually. You're just going to have to take my word for it, that degrading is better than inhibiting. So BCL6 is mutated in up to 85% of DLBCL, so there is a genetic link, which is a subset of non-Hodgkin's lymphoma. Obviously, a high unmet medical need, 18,000 people diagnosed each year. And there aren't many oral therapies for DLBCL. There's a lot of chemotherapy, there's androgen drug conjugates, there's these CAR Ts, but no oral therapies, which is obviously much more convenient for the patient. And our compound is oral. And then beyond DLBCL, there's Burkitt's lymphoma, follicular lymphoma, there's AITL, and also some solid tumors. So a big unmet medical need, a big opportunity for a BCL6 degrader. And we have a good one. So here's some data. This is an in vivo xenograft model using a DLBCL model. And you can see that with a low oral dose, so 3 mg per kg, 10 mg per kg, those are low. This is a quantitation of the amount of protein BCL6 that's left after that dosing. You can see basically protein goes basically down to 0 at both doses. So we're completely eliminating BCL6 from these cells, which is exactly what we want to do with the PROTAC. And the result of that is that these tumors stop growing, completely flatline. Here's the vehicle, so this is basically a water control. The tumors are growing quite rapidly, but with the BCL6 PROTAC, those tumors aren't growing at all. So the tumor stasis, 100% tumor inhibition, correlates with the amount of degradation. We're degrading 100%, we're stopping growth of the cells 100%. And we've seen that in multiple DLBCL models, which is obviously we want to have a breadth of efficacy here. And this program is currently in what's known as GLP toxicity studies, which is the last step essentially before you file your investigational new drug application and get into the clinic. And we expect that to happen in the back half of this year. So we're really excited about this program because it does have advantages showing degrading is better than inhibiting. There is an unmet medical need, and it's an oral therapy, which is really needed in this space, especially for some of these patients who are really late line, they're really beaten up. Oral therapies are much, much easier on them. So that's oncology. The next program up in oncology, we also have a big neuroscience portfolio. John alluded to it earlier. And we think that neurodegenerative diseases, neurological disease in general, is a big opportunity for PROTACs. We really think it can revolutionize the space because of the way the mechanism works. And obviously, there's been a lot of failures in neuroscience with the various modalities that are in play now, and we think we have an advantage with PROTACs. So we have PROTACs that obviously got to cross the blood-brain barrier when you're talking about these neurodegenerative diseases. So we're talking about Alzheimer's, Parkinson's, and you'll hear from Adam about Huntington's later. These have to reach targets in the deep brain regions. I'm going to actually show you data that these PROTACs do. Obviously, degrading the disease-causing proteins. So our publicly announced proteins are tau, alpha-synuclein, and Huntington. And I'll be talking about LRRK2 in a second. And they can differentiate between the mutant and the wild-type proteins. And I won't steal Adam's thunder, but that's part of the deal with Huntington. You want to stay away from the wild-type version and only hit the mutant version that's disease-causing. And key, also oral delivery. So an oral compound that can cross the blood-brain barrier, get into deep brain regions, degrade the disease-causing proteins. You can imagine that we believe that has a significant potential advantage over other existing modalities, like antibodies, like antisense oligonucleotides, which you have to inject intrathecally or into the spine, and that's no fun, obviously, as opposed to taking a pill or even just an IV therapy. So the next program, our first program, really, the most advanced program in neuroscience is a protein called LRRK2, or leucine-rich repeat kinase 2. It's linked to Parkinson's disease. Obviously, Parkinson's disease is the most common, second most common neurodegenerative disease, 2.5 million in the US, Europe, and Japan. Obviously, a huge unmet medical need. There's no disease-modifying therapies approved for Parkinson's disease. There's some symptom-modifying, but no disease-modifying. And LRRK2 is tied to Parkinson's genetically. So familiar mutations, whoops, wrong one. Whoops. Sorry, hit the wrong button. So there are mutations in patients that Parkinson's disease that's genetic or genomically inherited. The most commonly mutated gene is LRRK2, actually. Now, it's only about 5% of those patients, but it's more than any other gene that's mutated. And in the sporadic version, LRRK2 is overexpressed by twofold increase in expression in these patients. So really strong linkage of LRRK2 to Parkinson's. And what is LRRK2? It's kinase, but it's also one of these scaffolding proteins that John mentioned. It's got a kinase function, also has a GTPase function. It's got other proteins binding to it that all contribute to the activity. So just inhibiting the kinase actually doesn't take care of all the function of the protein, but degrading it, eliminating it, would get rid of all of those functions, including the scaffolding function. And there's actually human genetics that supports that when you look at reduction of the protein versus kinase inhibition, you actually see much better activity by down-regulating it, for example, with an antisense. And really, you only have to degrade 50% of the protein, which is advantageous, to actually have that effect. So again, degrading is better than inhibiting in this case as well. And so what happens with LRRK2, so the mutations or the increased expression of LRRK2 lead to lysosomal dysfunction, so the cell's not able to degrade proteins like tau or synuclein that then aggregate and lead to the Lewy bodies, for example, in Parkinson's. That's a hallmark of the disease. There's neuroinflammation that occurs, which is bad. Mitochondrial damage, which is bad, which all leads to the loss of dopaminergic neurons. That's the hallmark of the disease in the brain. So speaking of the brain, we have an oral compound. It's also in GLP toxicity studies, so late stage research with an IND scheduled for the back half of the year. And here we've dosed monkeys, so non-human primates, with our LRRK2 PROTAC and looked at different regions of the brain, the cortex, striatum, and cerebellum, striatum being really deep in the brain. And you can see that at, again, low dose, 5 mg per kg, in each of those brain regions, you get significant degradation, 87, 87, 89% degradation of LRRK2 in the brain. We've actually seen this as well in mice and rats. So across species, we see really robust degradation of LRRK2 in the brain and in deep regions of the brain, which is key, with an oral compound. So again, going back to that original slide, oral, crossing the blood-brain barrier, getting into the deep regions, degrading the pathologic protein. We think this is going to be really dramatically different than kinase inhibitors that are in the space. Okay, so that's it for me for the next 2 programs coming up. I'm going to turn it over to Katie, who's going to talk about her program, and then we'll hear from Adam as well.
Thanks so much. So I am excited to get to tell you just a really short snippet about one of our oncology programs that is on a previously kind of historically considered undruggable target. Let's see if I can do this. And that is KRAS, which many of you are probably familiar with this. But if you're not, it is kind of a key oncogenic driver in many types of cancer. And it has been very challenging to drug. The industry has been trying for almost 4 decades. And so we were very interested to see if our PROTAC technology could tackle this protein, which is kind of considered one of the holy grails of oncology. So what does RAS do? Why is it so important to target? So the RAS proteins are essential for cell survival and proliferation, but they're really designed to just turn on briefly in response to external signals in the cell's environment and trigger kind of a growth, proliferation, survival response, but then they turn off. What happens in a lot of cancers is that there are point mutations that occur in RAS that lock the protein in a perpetually on state. And so it's just constantly signaling the cell to grow, divide, survive. And this has unfortunately led to many, many different incidences of cancer. And as I mentioned, the role of KRAS in particular in human cancer was discovered 40 years ago. So this has been a 4-decade-long process of trying to bring therapies targeted therapies against this oncogene. There are multiple different point mutations that occur pretty commonly in this protein, but the three most common are G12D, G12V, and G12C. The little bit of data I'm going to share with you today is all focused on G12D. And Arvinas has really committed to being able to drug multiple forms of this oncogene, and so we have programs targeting kind of all of the most common mutations. So just kind of getting to the unmet medical need, depending on what you look at, somewhere between 25 and 30% of all human cancer patients have a RAS mutation. So huge incidence, and it's most prevalent in pancreatic, colorectal, and lung cancer. And I just want to highlight especially in pancreatic cancer, which we all know is such a devastating disease. Upwards of 90% of patients with pancreatic cancer have a KRAS mutation. So it's known to be the primary driver in this disease. And so we really want to be able to provide a targeted therapy for these patients. And, as Ian mentioned and John and others have mentioned, we think deeply about why a degrader might have advantages over an inhibitor in this space. Ian has touched on several of them. I'll just mention for KRAS kind of a few additional things. So one is the scaffolding role. And so KRAS is a protein that exists almost exclusively in a multi-protein complex, which is a model of that complex is shown here, where KRAS is kind of this salmon-colored or coral-colored protein in the middle. And so it's part of this very large complex that exists at the cell membrane. And so you can imagine if you degrade KRAS versus simply kind of inhibiting one of its functions, that might have significant consequences for the cancer cell. And so that's one of the reasons. And then the second is we see with PROTACs that they can often have a very durable pharmacodynamic impact. And this is partly due to the catalytic activity that John introduced at the very beginning. And then for KRAS in particular, this protein has a pretty slow resynthesis rate. So when you degrade it, the cell does not make it back immediately. And we also see that often PROTACs are able to accumulate in the tumor, and so they have this very extended activity. And so again, this is different than an inhibitor mechanism where you have to kind of keep occupancy continually to have your effect, whereas for a PROTAC, we can degrade the protein and have a more extended effect. So I'm just going to show you one slide of in vitro data and one slide of in vivo data. So these are G12D selective PROTACs, and we have been able to optimize to highly potent compounds. So, in this panel on the left, we kind of just show a progression of tool compounds that we're just calling kind of A through D. We're able to very quickly kind of optimize to picomolar degraders that essentially eliminate KRAS from the cancer cell. And you can see that maybe more easily visually in this Western blot, where this is KRAS G12D, and you can see that the protein is essentially completely gone. And that exists with a downregulation of the signaling that KRAS propagates in the cell. Here we're looking at a marker called phospho-ERK. And so you can see as KRAS gets degraded, phospho-ERK also gets suppressed. And so that it will slow the cancer growth and hopefully eventually keep the cancer in check. And lastly, with these molecules, they're highly selective for this particular KRAS mutation. So on this last panel, we've just, we have a degradation assay where the G12D is in green, and then wild type KRAS, as well as some of the other common mutations are shown here, where we're really not degrading them at all. So this is a highly selective degrader. So the last thing I'm going to show you is some in vivo data, and here we really do see some direct evidence for the differential activity of a PROTAC. And so this is in a colorectal cancer in vivo model. And one thing that I just want to draw your attention to first is we're looking at our PROTAC D, we're calling it. And so if you look at the green curve versus the blue curve, now if I can, this is a little bit hard without the laser pointer, but the green curve is down here and the blue curve is up here, kind of growing similarly to vehicle. And both of these molecules have a KRAS warhead that is able to inhibit KRAS. The only difference is that this blue molecule can inhibit only, whereas this green molecule also has the ability to degrade. But it's a very small chemical change, so same properties. And so we see this enormous difference when we add the ability to degrade. So this was very exciting. And then the second thing I want to highlight with this data is that we are able to give very low doses. So this is 1 and 3 MPK. And we're able to dose them pretty intermittently. So once a week or once every other week, and essentially achieve total tumor stasis. So again, kind of demonstrating that extended PD effect that we were talking about. And so this makes us really excited about a PROTAC approach for KRAS. And so with that, I'm going to hand it to Adam, who's going to talk about one of our neuroscience.
Thank you. Hello, everybody. Excited to give an update on our Huntington's degrader program today. This is an early program in neurology that's really, building on the progress that was made in neurology with the LRRK2 project that Ian was talking about before. Some of the lessons learned there were really well taken in this project. We're super excited about the way it's going. I'll tell you a little bit about the indication, Huntington's disease. It's rare but not ultra rare. About population incidence globally is about 5 in 100,000. Dominantly inherited, progressive, fatal neurodegenerative disease. It's caused by a trinucleotide expansion in the first exon of a gene that encodes a protein called huntingtin. Patients have extra glutamine residues in the extreme N-terminus of this protein. I'll try the spotlight here. Oops. Already botched it. Oops. There it is. Okay. The extra glutamines make the protein misfold. The misfolded region is then cleaved off, which liberates the soluble version of a very small, very neurotoxic exon 1 fragment inside the cell. And that cell is really, that fragment is really a driver of a lot of the pathology underlying Huntington's disease. It also has the tendency to oligomerize and aggregate, and this really forms the pathologist's hallmark of the postmortem Huntington's brain, which is these intranuclear inclusion bodies that you can see here. They're somewhat similar to Lewy bodies that you see in Parkinson's disease. Taken grossly, you can see that the effects of Huntington's disease on the brain are really devastating. It's just a diffuse loss of gray matter throughout the brain, particularly in the striatal region where the disease originally arises as a disease of sort of control of complex motor patterns. Huntington's disease is kind of the yin to Parkinson's disease is yang, but it's, begins with the death of these striatal medium spiny neurons, which are GABAergic projection neurons from the striatum. You can see in the healthy patient, you have the caudate putamen here quite robust. The lateral ventricle is barely resolved right on the median edge of the slice here, whereas in the Huntington's patient, the death of all these neurons has caused this lateral ventricle to fully dilate. And that type of sort of catastrophic loss of neurons is at the end stage is really diffused throughout the brain. It's really just a catastrophic neurodegenerative condition that ends in death in all patients. So the medical need is totally unmet. There are no approved disease-modifying treatments. We aim to be the first small molecule. The most clinically advanced treatments are antisense oligonucleotides, as Ian mentioned, a very onerous procedure to administer these, intrathecal infusion, complicates the life of complicates the already complicated life of a Huntington's patient significantly. And the issue with these ASOs is they don't spare the wild type allele of the Huntington's protein, and the wild type allele of the protein is neuroprotective. Patients need the wild type allele. So what we think we can do with our PROTAC, and why we think this is such a good modality, is we aim to go in and selectively degrade just that mutant allele of the Huntington's protein, which drives the pathology of the disease, sparing the wild type allele, which is neuroprotective. And as I'll show, we've been able to make some pretty good progress doing that. We can degrade the wild type allele, the mutant allele in neurons, and we can get these into the brain. Oops. Okay, so putting on our preclinical drug hunting hat, what is the profile that we want in an early tool molecule as far as a allele selective Huntington degrader, right? This is a Huntington's mutant Huntington expression system here. This is one of our PROTACs. You see degrading nicely, clearing 60 or 70% of the mutant Huntington in this system, nice and potent with about a 70% of 70 picomolar DC50. What we want, in addition to allele selectivity, is to show that we're on target at Huntington and on mechanism at the ubiquitin proteasome system. And the way we can do that in a relatively simple way is just running pharmacological competition experiments. So as you can see in the red trace here, we're running an excess of the ligand that recruits the E3 ligase. When we saturate the binding sites on the E3 ligase, we autopharmacologically compete off the binding of the PROTAC to the E3 ligase as we do here. As you can see, when we run an excess of the Huntington binding ligand, which is shown in black here, we ought to saturate the binding sites on the target itself, Huntington, and right shift the degradation curve there as we do as well, right? Indicating that we're on target at Huntington's and we're on mechanism at the ubiquitin proteasome system. So check and check. And throughout it all, you can see wild type Huntington, the neuroprotective form of the protein, is essentially unmoved. And here's the visual, right? Here's mutant Huntington concentration-dependent degradation, and here's the wild type allele unchanged. So this is the profile of a molecule that would progress in our funnel. Again, we're pretty early. We're working on translation in mice now, but this is a molecule that's looking pretty good, and this is the type of thing that we like to see in our early preclinical profile. To triage these molecules for going into mice, we really want to see them degrade in a primary cell. The target cell type, obviously, for a neurology PROTAC is a neuron. To model that in kind of a scalable way, we took rat primary neurons, we packaged mutant Huntington in an adenovirus, and we can then titrate the amount of target protein that's expressed in the neurons based on titrating the MOI of the adenovirus. So we can really match the amount of target in our cell system with the amount of target that we might see in various different Huntington's mouse models. So this is a really nice scalable compound triage system that we use. You can see here, interestingly and notably, the blue line is the soluble form of mutant Huntington. This is a 7-day incubation experiment. You can see the DC50s well below 1 nanomolar. At this time point, we also are degrading the insoluble form of the protein. I remember I mentioned that it oligomerizes, and the oligomers form aggregates, and the aggregates are sort of the pathologist hallmark of the late stage of the disease. We have other data that I won't show here that shows that this is not a direct pharmacological effect on these insoluble aggregates. They're essentially pharmacologically inert via this mechanism. But what we're doing is cutting off the supply of those soluble Huntington monomers that eventually oligomerize and make these aggregates, right? So it's heartening to know that we're actually moving both forms of the mutant Huntington that are associated with the pathology of the disease. And this has implications for biomarker work and things like that down the line as well. A molecule that's active in this model would be one that would be suitable for moving forward into Huntington's mouse studies, which is basically the stage that we're at with the project right now. If I could figure out this pointer. Okay, so obviously, we're early, just working on the mouse translation piece. The early pharmacokinetic data is really encouraging. This is free drug in a C57 mouse brain. Obviously, the goal in dosing is to establish a nice margin over your known efficacious concentration from your in vitro assays, and then restate that in a mouse. Here, at Cmax, we have a really, really nice margin out to 8 hours. The margin is still looking pretty good. Because the in vitro profile of these molecules at this point is really nicely dialed in, we can fight kind of a one-front war. This is really just an ADME optimization phase of the project that we're in now. I won't say anything beyond, we're just looking to sort of broaden this exposure profile, because that really opens up some more options in doing pharmacodynamic studies in Huntington's mice. So, the project is early, but looking really good. Really excited about the modality in terms of the options for, treatment options that it opens up, getting away from intrathecally infused therapies. PROTACs, as Ian mentioned, are small molecules. Oral administration is always the goal. We've been able to achieve that in LRRK2, and we've been able to show already that we can get a lot of these, decent, meaningful concentrations of these PROTACs across the blood-brain barrier. So we're excited for what's going to come next. And with that, I'll turn it back over to Don. There you go.
Thank you. So hopefully in the course of 50 minutes or so, you'll have at least had a snapshot of the company, the technology, our late-stage assets, and where we're going with the rest of the pipeline. A lot of exciting targets, very novel mechanisms, both in oncology and in neuroscience. And the next 2 years are going to be quite busy. Over this next period of time, we expect 4 pivotal trials to be ongoing. We have 2 that already started. There'll be another 2, both in breast and prostate cancer. We should have over 5 clinical trial readouts through that period of time, with at least one of these pivotal trials reading out towards the end of next year. And then we'll have 4 first-in-human studies from these new programs over oncology and neuroscience. So a very exciting time for the company. And I'm glad that you were able to listen to this, and thank you. And for whatever time we've got left, I'm happy to answer any questions. So thank you very much.
Sounds very, very impressive. Still got $1 billion of cash on the balance sheet.
How do you know that?
They tend to watch the quarterly balance sheet. So what are the stock analysts saying? We all know the challenges with publicly held biotech valuations right now. But,
Right.
what are they saying to you, and what are your shareholders saying? Because obviously, in the absence of data, things kind of drift a little bit.
Right.
Right. And you've got a lot of data coming. But what are you hearing, and what are shareholders saying relative to the stock price and your ability to raise capital should you need to over the next 20
That's a great question. I'll start, and Randy Teal, our head of corporate strategy, can add any components to that. But clearly, the market over the last 2 or 3 years has been rough for biotech, really tough. You're seeing companies going to the wall. You're seeing companies struggle to get that next round of cash, so they're downsizing as well. We were in the great position of having, just before COVID, $600 million in the bank, because we'd done a raise on the back of really good data. Then we did the deal with Pfizer, and that brought in another $1 billion. So we entered into this really bad period for the biotech industry with, over $1.5 billion. So it's allowed us to move forward. It's given us a huge amount of flexibility. But you see the pipeline we have. A lot of phase 3 pivotal trials coming up. We have a very robust pipeline. Nothing is failing, which normally is a really good thing. Literally nothing in our pipeline is stopping and failing. But it means we have to fund all of it. So there's a couple of different things. One is, I think investors do look to see what that next set of data is going to be. I think they're encouraged by the fact that we have a whole series of potential significant readouts coming up over the next year and 2 years. We also have the potential, because of this pipeline, to look at partnering. We did that great deal with Pfizer on 471. We do have the ability to do a similar type of question and say, if we had a strategic partner for any one of these assets, would it go faster? Would it be more comprehensive? So we have that ability as well. So I think, yes, the market's tough. The normal thing of showing good data, your stock price goes up and you're out raising, that model's not been working that well recently. But we do have a lot of confidence in the programs we have, and we still have a lot of financial flexibility. And I think that ability to partner gives us a lot of strength. Anything you'd add, Randy?
Yeah, and you touched on this a little bit, right? So we have a good problem, which is that we have pivotal data coming towards the end of next year.
Right.
So the tough thing is that it's hard to get anybody to focus on anything else. So with that milestone coming, we can show data on Huntington's and KRAS and all sorts of things, and that's the plan, right? We'll keep talking about more and more as they come along. If we rewind 6 months, we hadn't announced LRRK2 and BCL6 as our target. So now we're making a good solid point of getting out there to talk about what's coming next. At the same time, though, we do have to keep focused on the pivotal data that's coming. You know, John's talked about more pivotal trials starting at the second half of the year. So there's lots coming. It's just hard to get people to focus on things besides the top line, pivotal data coming.
Thank you.
Thanks.
I think another question here.
A great talk, more about biology, if I can ask. How specific do the target need to be on the proteins as compared to standard inhibitor?
Ian, do you want to talk about the specificity of PROTACs versus inhibitors?
Yeah, so we really try to dial in specificity, obviously. Adam with mutant versus wild type, Katie with the mutant versus wild type as well, mutant versus other mutants. Oftentimes the PROTACs are more selective than the warheads, which are often built on inhibitors from which they're derived. And that's because of the trimer complex. That extra layer of having to form a trimer complex, ubiquitinate on a lysine, and then lead to degradation. So, not every target do you need to have exquisite selectivity like we have in those programs. But in most cases we do. For example, the androgen receptor and the estrogen receptor, we've done proteomics experiments, right? So we've dosed cells, ground them up, run them through a mass spec, what's downregulated. It's only the androgen receptor, it's only the estrogen receptor, which is what we want. So that's basically our fundamental principle. We really want to be selective. PROTAC mechanism guides toward more selectivity than inhibitors, particularly with kinase inhibitors, which can be very dirty, when it's warranted. Not always warranted, but oftentimes it is. So PROTAC really lends itself very well to specificity selectivity.
How long is the cash forecasted to last you, given 4 late-stage clinical trials?
Yeah, if we do everything that we've laid out there, it's money through 2026.
2026?
Yeah.
Really?
Yeah, if we do everything. So it still gives us a lot of flexibility in terms of decisions about what to do, and as I say, partnering and also getting data that allows us to go out and raise cash.
And hard to forecast, but do you have any sense of when you might be filing a new drug application?
We haven't given that guidance yet. But you can see we've got late-stage assets that in the 471 space that are, we'll have pivotal data by the end of next year. So it'll be pretty close after that.
Thank you.
Well, great. Thanks very much for your time. Thank you very much.
Thank you. Absolutely.
That was great. Thanks.
Good job, everybody.
CAS.
Oh, thank you very much.
Nice job.
Are you all looking at universities? And have analog to other
What's been disclosed is Huntington's now.
Thank you. Good seeing you guys.
Yeah, we should evaluate on a conscious basis.
Yeah. Yeah.
Take care.
Oh, great.
We have a lot of good, but we also recognize that.
Yes, just a little bit.
Yeah, great. Yeah. Thank you very much.
Hey,
We've got
I'm here to
new CEO as well, CT, which is great.
Oh, right, yes.
Yeah, we need to.
Yes.
That's great. I mean, in terms of, even the second step,
Really? So you see the transition.
Oh, yeah, there's been a lot of transition. It's actually really good.
And lots of other.
There's more contacts, more companies. Biosynthesis have the ability to influence that.
But it's similar to.
Yeah, and I'm happy to be part of it, and you know, especially as it continues to grow. And the spice industry is getting larger.
It really is.
It really is, especially.
Yeah.
It's becoming, and it's obviously a big part of that.
Yeah. But we're glad we, I mean, most of the people here, they all had jobs in genetically in other companies. And so when we formed Arvinas, it was basically just to cool people. And sometimes those companies are leaving the state. Bristol Myers Squibb left the state.
Even if they're going to states, you know, it's kind of it's nice to be able to have that depth here.
I think again, I like
There's really not much like it. I live in Massachusetts.
right.
I have to say, I live in New Haven.
It's a little bit private situation. It's a nice shoreline.